Hi guys, so welcome back to my channel. So today we are going to look at subtopic 11.2 Dihybrid Cross, still under the chapter of inheritance, chapter 11. But before we go through in this lesson, I hope you are familiar with monohybrid cross. If not, then please go to my previous video to watch on monohybrid cross, then come back and learn on dihybrid cross. As usual, before we go through the lesson, let's look at a learning standard so that you know what you're going to learn in this lesson. And usually the questions are always around the learning standards. So firstly, you must be able to describe the meaning of a dihybrid cross and then explain a dihybrid cross based on Mendel's experiments, still using the P plan. All right. Then you should be able to construct a dihybrid cross diagram, same like whatever you did in monohybrid. And then you are going to determine the genotypic and phenotypic ratio of filial generation, the F1 and F2 generation, in a dihybrid cross. And then you are supposed to be able to describe the Mendel's second law. Okay, so what is dihybrid inheritance? We know in biology, di meaning two. All right, so dihybrid inheritance is an inheritance that involves two characteristics. Okay, and each characteristic is controlled by a different gene that is located on different locus. So we have two genes, okay, that control specific characteristics. For example, we have, we want to talk about tall, okay, height. We will talk about height, so it's either tall and short, okay, or we talk about color of flower. So we have purple and also white. Okay, so these are the characteristics that we are going to see. So in monohybrid, you see only one characteristic, is either a height, but in dihybrid, you can also see the characteristics. For example, we have height and also flower color. All right. Okay, so genetic crosses in a dihybrid cross involves two different characteristics. Like I said just now, it's either seed shape or seed color or any other characteristics that we want to crossbreed and see. And it involves a pure breed. Okay, the parental generation are both pure breed for both the characteristics. So one will be either homozygous dominant and the other will be homozygous recessive. For example, we have the height just now, height and flower color. All right, so here for example, homozygous dominant, so let's say height tall and flower color will be purple, so this is homozygous dominant. And homozygous recessive, so the height will be dwarf, so small t, and recessive for color, flower color, small p. All right, so these are pure breed and it involves two characteristics in dihybrid cross. Okay, so let's look at Mendel's dihybrid cross. Okay, in this experiment, Mendel carried out genetic crosses which involves two different characteristics of a pea plant, which is seed shape and also seed color. So Mendel crossed the pure breed of the plant, okay, which has round yellow seed. So both traits are homozygous dominant. All right, with a pure breed of pea plant that has wrinkled green seed or constricted green seeds, and both traits are homozygous recessive. Okay, from his experiment, Mendel obtained the following results. Okay, that means all offspring of the F1 generation, the first filial generation, has round and yellow seeds. Okay, so this is heterozygous, okay, genotype, heterozygous genotype, but all are round and yellow. That means the dominant traits is still intact. Then the self-pollination of the F1 hybrid, okay, will produce F2 plants, okay, F2 generation with the following genotype ratio, which is 9 to 3 to 3 to 1. That means 9 round and yellow seed, 3 round and green seed, 3 wrinkle or constricted and yellow seed, and 1 wrinkle and green seed. Okay, so how do we get that? Okay, from here, I repeat again. Okay, so here we have purebred, okay, dominant and recessive over here. So we take out the gamete. When after they undergo meiosis, both characteristics have to be shown, okay, in the gamete. So B, capital B is for round seed, and capital K here will be for yellow seed. Here small b will be for constricted seed, and small k will be for green seed. So here we have, when we fertilize them, random fertilization happen. So we get round and also yellow seed. Okay, so this round and yellow seed, if we have to cross pollinate them, okay, during meiosis, we will produce this. How do we get this? Is actually we are cross pollinating the characteristics that, or the alleles that is in the F1 genotype. For example, we have B, B, K, and K. So what happens here is, how do we get this? Is by crossing this, okay, between the two characteristics. So we have capital B, capital K. Then this B and this B and the small k. So we get capital B, small k, and the small b with this capital K, you get B and capital K, and the small b with the small k, you get B and K. So that is why we get this. Okay, so we get these gametes. Okay, same goes for the second parental chromosomes. Okay, parental genotype. So now here, when they undergo fertilization, okay, we definitely have to use the Punnett square. Okay, how do we use the Punnett square? Is from here you can have a look. Okay, so all the gametes, okay, from the female and from the male is actually the same thing. Okay, but when we cross them, you will get different results. Okay, so this and this, you get round and yellow. This is dominant, homozygous dominant. Okay, here you get homozygous dominant for round seed and heterozygous for yellow seed. Then this one and this one again here. Okay, so you use the Punnett square. You cross like this. You cross here one row by one row. So here you cross, you get B, K and BK here. You get round heterozygous and yellow homozygous dominant. Okay, and then we have here is both heterozygous, but still the dominant trait is shown. Okay, so same goes with the rest. So you can cross them. Okay, you can cross each, each of these. All right, and you will get the ratio 9:3:3:1. That means you will get 9 round and yellow seed. Doesn't matter whether it is dominant, whether it is heterozygous. Okay, so you will get what is this called? 9 round and yellow seeds. Okay, and then we have 3 round and green seed, 3 constricted and yellow seed. Round and green meaning is can be round, can be homozygous, or the round can be heterozygous. Okay, and green seed, since it's recessive, so we have both small k. Okay, from the Punnett square. Constricted, on the other hand, constricted is recessive, so we have small b, definitely small b. Yellow seed can either be dominant or it can be heterozygous. And constricted and green seed, these are both recessive, so the only possible genotype will be small b and small y. Okay, so from here, this is all we get from the diagram, from the Punnett square. Same concept again. Okay, here again, round and yellow seed, same thing. Okay, if you can't understand my handwriting just now, okay, let's look at this. Okay, so the parental genotype, we have round and yellow, wrinkle and green. That means it's homozygous, okay, dominant, and homozygous recessive. Okay, undergo meiosis, they produce gamete with both characteristics still intact. Okay, and the F1 genotype will be all round and yellow seed, but they are now heterozygous. Okay, heterozygous. So, and these possible gametes after we self-pollinate the F1 generation. So you will get, okay, R and Y over here, and this one over here. Same goes with this, R and Y, this one, both characteristics. You have to cross them first to get the gametes, and then these gametes you put into the Punnett square. Okay, you put into the Punnett square and then you cross that. For example, in this case, if we want to cross this and this, it comes over here. So we have RR, YY. So we have wrinkle and also yellow seed. So we will come up with we'll come up here with a Punnett square. Okay, so these are done. Okay, these are done. So you can try it out. So when you count it, this is round and yellow, round and yellow, round yellow, round yellow. This is also round yellow. This is round and green. Okay, this is round yellow. This is round and green. Okay, this is round yellow. This is also round yellow. So we have about 1, 3, 4, 5, 6, 7, 8, and this is also round and yellow. So we have 9 over here. Okay, then we have, let me use another color. This is round and green. Okay, this is also round and green. There's any other round and green? Yeah, this is also round and green. So we have 3 round and green. And then we have wrinkle and yellow, wrinkle and yellow. Oh, sorry, another color. Wrinkle and yellow, wrinkle and yellow. And this is also wrinkle and yellow, which is also 3. And the last one we have over here, this is wrinkle and green, which is 1. So from there we get the phenotypic ratio 9:3:3:1. Okay. So remember, in monohybrid cross, okay, the phenotypic ratio is 3:1, okay, and genotypic ratio is 1:1, 1:1. And in dihybrid cross, okay, since it uses 2 characteristics, okay, so we use only the phenotypic ratio, which is 9:3:3:1. Okay, percentage you can calculate by using the by calculating the total phenotypes that is calculated. Okay, so based on his experiment on dihybrid cross, Mendel proposed that Mendel's second law, or we call it as law of independent assortment. It says that during gamete formation, each allele from a pair of alleles can combine randomly with any allele from another pair of alleles. This means that during gamete formation, alright, the segregation of alleles for seed shape does not affect the segregation of alleles for seed color. Alright, and the parent has to be pure breed, parental generation. Then the alleles for each characteristics, okay, they segregate independently of one another, which results in the production of gametes with different allele combination. And each gamete will receive one allele, okay, from each pair for each characteristics. And due to the segregation of allele, each allele from a pair of allele for characteristics may combine randomly with either allele from another pair. For example, in genotype here we have here BBKK, BBKK, and then first filial generation here, so it can actually combine. So our combination is our gamete formation here. Okay, it receives one allele from each pair of characteristics. One is from the seed shape and the other is from the color of the seed. So we have both B and K over here. Okay. Alright, so there is a question over here, okay, to talk about dihybrid cross. Now, a dihybrid cross between 2 varieties of palm oil, or oil palm, Dura and Piscifera, produces a new variety which is called Tenera. Tenera has better quality fruits compared to Dura and Piscifera. Now, self-crossing between Tenera varieties does not produce offspring of the same quality as the parents. Why? Okay, so we have here H is a dominant allele for thick husk and F is dominant allele for thick flesh. So the opposite will be small h and small f. So now Dura and Piscifera are parents. So they are parental generation and we can see they are pure breed over here. Okay, they are homozygous, either dominant or recessive. So if we cross them, use another color of pen. Okay, so if we cross them, okay, so we have here Dura, okay, FF, and we have Piscifera, HHFF. By the way, it's actually to produce Tenera, which is a better quality compared to the parent. Alright, so when we cross them, so here we have H and F. This is the gamete form after meiosis. This is capital H and small f. So random fertilization happens over here. So it produces the first filial generation with all thick husk and thick flesh. So Ternera is this. Ternera's genotype will be HHFF. Alright, so this Ternera is the F1 generation. But now, if Ternera we cross with another Ternera species, we definitely will not get, okay, the same characteristics. So here we have to use the Dihybrid Cross. Change this, please. Okay, from the question just now, let me just put it back again. So we have Dura and also Piscifera, HHFF. Okay, so after meiosis, the gamete we have here is HF, and here we have this. All right, and then when they undergo fertilization, we produce HHFF. Okay, so this is the genotype. Phenotype will be all thick husk and flesh. Okay, so you can write it yourself. I do not have enough space over here. My handwriting is too big. Okay, so we have this. Okay, this is the first filial phenotype and genotype. Okay, now from here we want to cross now. Okay, phenotype is the same. So genotype is here HHFF. We cross pollinate them again H, F, and F. Okay, so the gamete we are going to have here is 4 different gamete. Okay, sorted independently. So we have here HF, and then we also have this one, and then we have small h, big F, and also small h and F. Same goes with this. Okay, we have the dominant, and then the dominant and recessive, dominant, and recessive. Okay, so these are the possible gametes after undergoing meiosis. Now, after fertilization, we use the Punnett Square. Okay, we use the Punnett Square. We can actually get ratio of 9, 3, 3, 1. Okay, so that's how you draw a schematic diagram. Of course, this is not complete. Okay, you can draw a complete schematic diagram based on what you have learned. Okay. Okay, so we are done with that. Now you can do formative practice 11.2. Okay, so here the answers will be given in the description box below. So with that, I will end my video here. Thank you for watching, and please do not forget to like and subscribe.